Welcome, everyone. My name is Yishan Zheng. I'm director of Carnegie's Department of Embryology. Thank you for joining us for our fall Science in the Neighborhood public lecture. Today, you will hear from a leading scientist, Dr. Stephen McKnight, a former faculty of our department and currently a professor at UT Southwestern Medical Center. Steve will describe his research concerning a class of proteins that do not form stably folded structures and their roles in neurological conditions that are pretty familiar to many of us. My colleague, Dr. Joseph Gall, will introduce Steve. Joe is a numinary biologist who has made great contributions to our understanding of many aspects of cells, which are building blocks of organisms, including ourselves. For example, Joe pioneered the study of chromosome ends called telomeres and many other structures in the cell's nucleus. Some of these structures contain the type of proteins that our featured speaker will describe further. Joe is often regarded as the founder of modern cell biology and was recognized by many prizes, including the prestigious Albert Lasker Special Achievement Award in 2006. Okay, Joe, you can now take away. Thanks.
It gives me great pleasure for many reasons to introduce Steve McKnight as speaker in today's Science in the Neighborhood public lecture. Steve's career and mine have intersected at many points, beginning when he carried out his graduate work at the University of Virginia with Oscar Miller, who had been my first postdoc. Then he came here to the Carnegie, first as a staff associate in 1977, and subsequently in 1983, when he returned as a staff member. That same year, I came to Carnegie as a staff member, so we had many interactions because of our shared scientific interests. He left Carnegie in 1992 and co-founded his first company, Tularic. He returned to academic research in 1996 as chairman of the Department of Biochemistry at the University of Texas Southwestern Medical Center, where he has been ever since. His research has focused on numerous aspects of cell and molecular biology. Today, we welcome Steve back to Carnegie to present his lecture entitled, How Do Gibberish Low Complexity Domain Proteins Work? The platform is yours, Steve.
Let me start by thanking both Yishan and Joe for the kind introduction. As they mentioned, I was a Carnegie scientist. I got there in the fall of 1977, so that's 44 years ago. And more or less, my first 15 years of science were all Carnegie. And I'll always be indebted to the Carnegie Institution of Washington, and in particular, the Department of Embryology, for betting on me. And so to be able to come back and talk about my science late in my career at this point is a fantastic honor. All right, let me get into the science. So what I'm going to talk today about is two longstanding enigmas, and I'll get to those in a second. But what I want the take-home lessons to be are spelled out in this slide. So first of all, proteins of low sequence complexity can transiently adopt molecular structure in a facultative manner. And what I mean by facultative is that protein structure occurs optionally in response to circumstances. So these low complexity domain proteins don't fold into a permanent, stable structure. They go in and out of their structural state. And that process of going in and out of the structural state can be easily regulated. And the second point is that this fluctuation in protein folding, this in and out of adopting molecular structure, we think may relate to the dynamic organization of nucleoplasm and cytoplasm in eukaryotic cells, and in particular, puncta within the nucleus and cytoplasm that are not surrounded by investing membranes. And these are puncta that have been known about for many, many, many years. And in fact, Joe Gall studied intensely for the last 10 to 20 years of his own career. And the final point I want to make is that neurological diseases often result from mutations that can enhance the stability of these otherwise labile structures, the structures that are formed from these low complexity domains. So those are the points that I would like to get across in today's seminar. So let me now go to kind of explain the two enigmas, the longstanding enigmas. The first is the enigma of what we call low complexity sequences. And this goes back to the 1980s when we were first successful in purifying transcription factors. And that's something I did while I was at the Carnegie, and as did many other people, Bob Rader, Bob Tejan, and others. So we were trying to understand how gene-specific transcription factors worked. And on this slide, you see a prototypic gene-specific transcription factor. And we came to realize that they're composed of two domains. So one domain is shown over here on the left, and that would demark the DNA binding domain of a transcription factor. And those domains adopted normal structure. They adopted structures with alpha helices and beta pleated sheets in a stable manner. And the shape of those proteins allowed the DNA binding domains to specifically recognize DNA so that a transcription factor could sit on the right piece of DNA and regulate the appropriate battery of genes. The second thing that was unexpected is that we found that these transcription factors had regulatory domains. And the earliest ones that we studied, we called activation domains. And those are shown on the right in blue, the squiggle. And what they did is they somehow tickled the transcription machinery to activate gene expression. So the DNA binding domain of a transcription factor was inadequate to regulate gene expression. It could bind to the right genes, but it needed this auxiliary domain, this activation domain, in order to trigger gene expression. And so that was interesting. We didn't know what to think of it. These were new. So, we wanted to study how they worked. And that was problematic because the activation domains did not assume molecular order. They did not fold into a stable shape. And yet they were functional. They worked, but we couldn't sort out how they worked. And the principal reason that we couldn't sort out how they worked is they did not adopt a specific molecular shape. So this was maybe 30, 35 years ago that we stumbled over these. And to this day, it's still enigmatic as to understand how activation domains work. Now, when I say they're of low sequence complexity, what do I mean by that? What I mean is they don't use the normal distribution of 20 amino acids that a typical protein would use and need to fold into three-dimensional shape. So what was noticed about these activation domains is that they would be composed of only a subset of the 20 amino acids, maybe 2 or 3. So Bob Tejan studied the activation domain of the SP1 transcription factor, and it consisted of like 30 or 35 straight glutamines. Well, that's low sequence complexity. It's not playing with the whole deck. And it was not surprising to see that those proteins could not fold because they didn't have the proper distribution of amino acid residues necessary for folding. The protein I studied was the VP16 protein. It was also of low sequence complexity. It was acidic. It had a number of aspartic acid and glutamic acid residues. But again, it was of low sequence complexity. It would not fold into a specific shape. And so we couldn't really figure out how it worked. So low complexity sequences have long been enigmatic. So whereas they were discovered in the context of transcription activation domains, it's turned out that these are all over the place. They're in transcription factors, they're on RNA binding proteins, they're the FG repeats in the permeability channel of nucleopores. They're on either end of all 75 of our intermediate filament proteins, keratin and the like. They're in all the subunits, almost all of the subunits of the mediator complex. They're histone tails of low complexity. So they're all over the place in cell biology. They're not relegated solely to transcription activation domains. So these are widespread. And there may be 10 to 20% of our proteins are of this nature. So they're a big unknown in biology. The other enigma, the second enigma upon which this research program has kind of been based, is the enigma of puncta within cells and embryos and neurons that are not surrounded by bilayer membranes, lipid bilayer membranes. So these are, again, all over the place in biology. On the left, I show a C. elegans embryo with its germ granules, and studied beautifully by Geraldine Sadeo at Hopkins Medical School and many, many other people, Susan Strom, and I could go on and on. So what are these polar granules? What do they do? How are they organized? If they don't have a membrane around them, like a mitochondrion or a peroxisome, what organizes them? What brings them together? What causes them to be only on the posterior side of the embryo, as shown here? So lots of questions. These are all over the place. The second example I give is neuronal granules, and they contain messenger RNA and protein. Of course, the messenger RNAs are made in the nucleus of the neuron, but the messenger RNAs get exported out of the nucleus and out of the cell body and move down the appendages of nerve cells for localized translation at active synapses. And so how does that happen? What holds these particles together? How do they know to be translated at the appropriate place in the vicinity of an active synapse? The next example is stress granules. So if you look at mammalian cells and heat shock them or deprive them of oxygen, they lead to the formation of these puncta within cells that are very clear cut, but no membrane around them. How are they organized? And finally, on the right-hand side, I show processing bodies in yeast. These have been studied by Roy Parker and many other terrific scientists. If you deprive Baker's yeast cells of glucose, you see very prominent processing bodies. And again, what's the organizational principle holding these together? So my argument is going to be that the low complexity sequences are at the heart of this organization. And that is a speculative conclusion, but I'm going to start with that because these are the two enigmas that have framed our research over the last decade. All right, so there are three parts to the seminar today. The first is, how did we get started on these low complexity sequences and our adventure into this phenomenon of protein phase separation. The middle part will discuss our more recent work on an RNA binding protein called TDP43 and in studies of how its low complexity domain might work. And the final part of the seminar will address the question, how does the addition of a single hydrogen bond profoundly change the behavior of this TDP43 LC domain? Also, we've found the same thing in the HNRPA2 protein, the tau protein, which is so important to neurodegenerative disease, and neurofilament proteins. So the first part of the lecture will be all published data. The middle part will be some published data and some new things that have not yet been published. And the final part will all be new data that have yet to be published. All right, so the first part of the seminar, phase separation of low complexity domains. How did this happen? So about 10 years ago, Masato Kato in my lab and Shinlin Du and others were working on the amino terminal region of a protein called fused sarcoma, or FUS. So it's a low complexity domain, and don't ask me why we were studying it, but we were studying that region of protein, and kind of noticed that the solution got cloudy, and in the microscope, what happened is droplets formed, and if that solution was incubated for a prolonged period of time, the droplets matured into a gel-like state, which is shown on the right-hand side of part of the slide. And I'd never seen behavior like this. I'd never seen a protein form a gel. And so here we have, these protein domains of low complexity sequence. And I thought, geez, this is interesting. Let's pursue it. Let's dig into this. And so that formed the basis of kind of our work for the last decade. What we've wanted to do is to study this process of phase separation and the formation of gels in hopes that these represent biochemical assays for the function of low complexity domains. Part of the reason that we failed to understand how activation domains work is we had no biochemical characterization that would allow us to study it. So here I thought, well, these processes of phase separation might represent the biochemical assays that would let us dig in and do reductionist science to perhaps understand how low complexity domains might function. So, we've used these over the last 10 years, and sure enough, they've really helped us. So kind of the question is, what's the basis of phase separation? What's present in these droplets and in gels? And so I more or less did what I was taught by Oscar Miller and Joe Gall, and simply looked. So on the gel, the hydrogel on the right, all we did was to put an electron microscope grid right onto the gel surface, take it off, negative stain it, and look to see in the EM what it might be. And what we saw was that the gel was composed of unbranched amyloid-like polymers. So as you can see in the EM image on the left, you can see these long unbranched polymers. They're beautiful, clean, uniform morphology. And so clearly the gel contains polymers. We also lyophilized the gel and put it in the X-ray beam and looked in the X-ray beam and noticed that there were strong diffraction rings at 4.6 to 4.7 angstroms, the outer ring, and 10 angstroms. And the combination of the electron microscopy and the X-ray diffraction immediately told us what we had. And that was, we had some form of amyloid polymer. And let me go on the next slide to the spectacular work of Linus Pauling in the early 1950s that told us about beta pleated sheets. And so the way that beta pleated sheets are held together is by this network of hydrogen bonds that holds two polypeptide sheets together. And those are, the distance between the two polypeptides is 4.7 angstroms. So that's the diagnostic reflection that we saw in the X-ray beam. And so sure enough, what we're dealing with is amyloid-like polymers. Now, let me go back 2 slides to one that I skipped and give you kind of the reason that we thought it was worthwhile to pursue these unusual observations. So what we wanted to know is whether there was any relationship of fuse phase separation to RNA granule formation. And what I've neglected to mention is the fact that fuse is a resident protein in many RNA granules. And so our idea was that maybe the role of fuse in these RNA granules would in some way be related to the phenomenon of phase separation that we were observing. And so the first point that kind of gave us some courage to proceed was that there was a concordant behavior of tyrosine mutations. Tyrosine mutations that prevented fuse from forming hydrogels perfectly matched those that impeded fuse from going into RNA granules in living cells. So there was a concordance there that gave us some courage. There are 27 tyrosines, and they're part of the low complexity of the fuse LC domain. There are a huge number of tyrosines, and those are somehow important for both phase separation and granule formation. The second part that gave us some courage was that Masato and Tina, Hahn, and others did kind of a crazy experiment. They took hydrogels and incubated those gels with lysates and allowed the lysate to bind to the hydrogel and then purified the hydrogel away, washed it extensively, and then we did proteomic experiments and RNA-seq experiments to ask what sorts of proteins or messenger RNAs would adhere to these hydrogels. And what we observed was that the hydrogels would capture exactly the same RNAs and proteins that are known to be in RNA granules. So that the match of what the hydrogel would bind in the known constituents of these granules was near perfect. And so we thought we're fairly safe in proceeding with this kind of crazy science. All right. So together with Rob Tico, we went ahead and used solid-state NMR spectroscopy to solve the molecular structure of the amyloid polymers that were in the hydrogels. And what you're seeing in this slide is the cross beta core that forms this structure. And so kind of what's not shown to scale here is that there's another 37 amino acids that are not structured over here on the amino terminal side, and another 110 or 120 amino acids that are beyond this tyrosine residue 97. So the structure is formed by a small part of the low complexity domain. Much of the structure is dangling off on either end, but kind of right there in this 40 residue area, a real structure is formed. And it was a hard task. Rob is a terrific solid-state NMR spectroscopist at the NIH, and working with Rob, Masato, and Dylan Murray, we're able to solve this structure, but it took 5 years. That's the bad news. The good news is that now by cryo-EM, what took us 5 years can be done in a matter of months. And David Eisenberg and his colleagues at UCLA have solved 3 or 4 more of these cross beta core structures from LC domains. So I think in the ensuing years, you're going to see lots and lots of structures, and we're going to learn lots more about it because of the advances in technology. Now, here's where the typical science would throw it in the trash can. There's no way that you could imagine that good biology could come from cross beta amyloids. Their history is terrible. They have been implicated over and over and over in neurodegenerative disease. And so one could hardly possibly imagine any biological utility to the formation of a cross beta structure like this. But I didn't know enough to realize that this was a foolish idea to pursue. So we kept going. And we did the experiment shown on this slide. And this was quite important. So what we did is to run our polymers on an agarose gel in the presence of increasing amounts of SDS. And shown on the left is a gel where we ran the prion protein from yeast discovered by Reed Wickner. And this is the SUP35 prion protein. And what you can see is that the prion protein, even if you add 0.5%, 1%, or 2% SDS, the prion polymers stay intact. So those are interactions that are extremely stable. I won't call them irreversible, but they're exceedingly stable, and they're not melted by even 2% SDS. By contrast, our polymers, they melt upon dilution. So as soon as we put them into the well of the SDS gel, they're converted from the polymeric state down to the monomer state. So in essence, there's something about the polymers that we're studying, the cross-beta interactions that we're studying, that is radically different from the prototypic cross-beta pathogenic amyloid. And so the question is, what's the basis of the difference between the two? So on the next slide, I show kind of just a cross-section, one monomer from our polymers, the ones that we've studied with Rob Tico and Dylan Murray on the left, and a subunit, a cross-section of a subunit from polymers made from the alpha-synuclein protein that are disease-causing pathogenic polymers that are nearly irreversibly assembled. And so if you simply look at the squiggles of the polypeptide chain, the two of them look very, very similar. And so you can't see much difference, but when we inspect the location of hydrophobic amino acids, what you can see is that in the alpha-synuclein polymer, there's 24 hydrophobic amino acids at the polymer interface. By contrast, the fused polymers have a single hydrophobic residue, and it's actually a proline, which is not a terribly hydrophobic amino acid. So one radical difference between the two is that the alpha-synuclein polymers have lots of hydrophobic glue holding subunits together, and that's not the case for fused polymers. A second that I think equally important difference between the two is that our polymers, whenever we make fused polymers, they always make the same structure. We've done it over and over and over, and every time they adopt the exact same structure. In other words, they can be folded and unfolded, and when they refold, they follow the Christian-Anfinsen rules. They know how to fold into the right structure. This is not the case with pathogenic polymers. So alpha-synuclein or A-beta, when you make polymers or study polymers from the brain tissue of diseased patients, all the polymers are different. They don't know how to follow the Anfinsen rule. They don't know how to adopt the same structure over and over. And so I think the fact that the polymers that we're studying know how to fold into the right structure over and over will end up being of profound importance. All right, so let me give kind of, at this point, a synopsis of the last 8 or 9 years of our work on phase separation. So these low complexity domains can self-associate so as to cause phase separation. Other people have done that, so that's correct. Phase separation assays are useful for performing reductionist biochemical, molecular biological, and biophysical experiments. What we've done is to use these assays of phase separation, as biochemical assays. We don't know what they mean with respect to the broader challenge of phase separation in cells. We hope that by studying them in a reductionist way, we might learn something, but in reality, all our focus is on using phase separation as an assay to study what's going on. The third point, self-association is mediated via the formation of weak and transient cross-beta interactions limited to a small part of the LC domain. As I told you, the structure that we found in fuse is only a small part of the LC domain. It's not the entire thing. And a structure is formed, but that structure is weak and transient. It's not like pathogenic, irreversible polymers that have been studied in the context of disease, neurodegenerative disease. And the final point is that self-association of LC domains is highly specific and readily controlled by post-translational modification. And I've not shown you the data indicative of those 2 points, but we know that these structures that are formed are extremely specific, that the fuse polymer can fit a subunit of itself onto itself, but it can't take any other LC domain and put it on. So it's a very specific reaction, and it's easily controlled by phosphorylation. And we published that, and I'm not going to show you the data. All right, now I'm going to move to TDP43. So what is TAR binding protein 43, TDP43? It's an RNA binding protein that many people have studied, and the most important work on it was done by Virginia Lee. And she did kind of a crazy inspired experiment. She took the brain tissue of diseased patients, people with terrible neurodegenerative disease, and simply looked for insoluble proteins. There was not a genetic trace that led her along this pathway. She simply looked for proteins that would be present in the brain tissue in an aggregated state, and she found TDP43. And it's turned out that TDP43 is extremely important. Subsequent genetic studies have shown that any of a number of mutations in the TDP43 protein can cause it to aggregate and lead to neurodegenerative disease. So TDP43 is an RNA binding protein, and we've begun to study it over the last several years because of a unique feature that I think it may end up being quite important. So, all right, where is it? So if you stain for TDP43 in cells, it's primarily in the nucleus. And as shown on this slide, you can see that it's in a punctate pattern in nuclei. So that's the most prominent thing observed when you just look at the bulk of TDP43. But in reality, it shuttles in and out of the nucleus to carry messenger RNAs to sites of translation in the cytoplasm. So it's not a permanent resident of nuclei, but it goes in and out of cells to facilitate appropriately localized translation. And one of the most interesting places that it's been studied is in the context of nerve cells. And as I mentioned earlier, nerve cells, transcription, of course, and the production of RNAs, including messenger RNAs, occurs in the nucleus of the cell, but they have to get out of the cell, messenger RNAs, to be translated in the cytoplasm. And TDP43 is one of a number of different RNA binding proteins that coalesce messenger RNAs into messenger RNP particles that turn into RNA granules. And then those RNA granules can move up and down dendritic appendages of neurons for appropriately localized translation. And so in the context of neurobiology, people have found that localized translation tends to occur very selectively at the sites of active synapses. And so a really exciting mystery is if you have a granule with TDP43 and other RNA binding proteins moving up and down an appendage, how does it come to be translated specifically at an active synapse? So TDP43 is a RNA binding protein. It has RNA binding motifs, but of course it has a low complexity domain. So our interest in it came from looking at the sequence of the TDP43 low complexity domain as a function of evolution. So here are the sequences of human TDP43 low complexity domain and chickens and octopus and all the way down to fish. And what we observed is that there are 10 methionine residues that are conserved through evolution. They're shown in yellow. And this piqued our interest because this was different from the other low complexity domains that we'd studied before. As I said, the fuse low complexity domain has 27 tyrosines. The H&RNP low complexity domain that we've studied also has lots of tyrosines. Other low complexity domains have lots of phenylalanines. So many of them have tyrosines and phenylalanines, but this particular one, instead of having an abundance of aromatic amino acids, had 10 conserved methionines. And so that struck us as being interested. So what we did is to carry out kind of our standard studies. You know, if we take the TDP43 low complexity domain, as shown over on the left, it will make these liquid-like droplets, very much like fuse. It will phase separate. And yet, because of the methionines, we thought maybe the methionines might be sensors for redox control or oxidation. So we tried a very simple experiment, and that is to simply titer in, as shown above the images, increasing amounts of hydrogen peroxide. And so H2O2 hydrogen peroxide did nothing to the fused LC domain liquid-like droplets, even up to 1% H2O2. And as shown on the SDS gel, that the H2O2 did oxidize the single methionine in fuse. You can see the two bands. And so the fused protein got oxidized, but nothing happened. By contrast, when we added H2O2 to the TDP-43 droplets, the droplets disappeared, they melted. And they melted as a function of oxidation. So you can see on this SDS gel, the protein that's fully reduced is down here at the bottom, but as it gets oxidized, its electrophoretic migration slows down. So at a certain point, maybe somewhere between 0.03 and 0.1%, the droplets melt. And we infer that that melting might be happening as a function of methionine oxidation. So sure enough, the H2O2 was oxidizing methionines, not surprisingly. And sure enough, this was reversible. And it was reversible by virtue of Earl Statman's two methionine sulfoxide reductase enzymes. So 30 years ago, Statman discovered two enzymes that can reverse and repair methionine oxidation and convert a methionine back to the reduced state. And so let's look down here on the bottom of the slide. So if we take our droplets and we melt them with hydrogen peroxide, if we then expose them to Earl Statman's enzymes, if we add both of the enzymes, A and B, we get the droplets back. They reform perfectly, and they reform because the Statman enzymes can reduce the methionine sulfoxide entity. And you need two of them, A and B, because each of them works on a different stereoisomer of methionine sulfoxide. So you need both. If you try and regenerate droplets with only one of the two enzymes, you kind of get halfway there. So you need both of them. But you not only need those two enzymes, but you also need thyroidoxin reductase, and NADPH, because you've got to move that oxidation potential downhill to the terminal conversion of NADPH to NADP. So if we're kind of watching the oxidation state, that it goes from H2O2 to the methionine residues, and then methionine residues to the enzymes, and the enzymes to thyroidoxin, and then thyroidoxin to thyroidoxin reductase, and eventually conversion of NADPH to NADP. So the important point here is this is a modification that's reversible. So the Statman enzymes are like phosphatases. So we all know that kinase reactions are not permanent because phosphatases can erase a phosphate. Well, the same can happen here. So this process of methionine oxidation is fully reversible. Well, that caused us to realize that maybe we could use methionine oxidation to probe the structure of the TDP-43 protein in this phase-separated state. And let's go to the next slide and kind of show what we did collaboratively with Ben Tu and Sina Gamagami. So Sina and Ben and Masato got together and did what I think is a really cool experiment. And that is they took our TDP-43 polymers and just sprinkled on a little bit of H2O2, a very small amount of H2O2 is kind of shown in red. And what we used initially, the red H2O2 is with the normal isotope oxygen-16. So here's what happens is that domains of TDP-43 that are not structured, these squiggles on either end, they're not in a structured state, so they're very easily oxidized. But in the middle of the structure, as exemplified by methionine number 2, the idea was that maybe that methionine would be protected from the structure and not initially get oxidized with the small amount of H2O2 being used to probe the polymers. after that reaction, then we could denature, we could denature and then hit it hard with H2O2, where the oxygen atom is the 18 isotope. So if this M2 methionine was not oxidized in the initial reaction, it would definitely be clobbered by a saturating amount of H2O2 O18. And so after doing these two reactions, we could trypsinize the sample and run a, do mass spec to look at the ratio of O16 to O18. And so if there's a lot of O16 and very little O18, then interpretation was that residue was not in the structure. By contrast, if there's a lot more O18 there than there is O16, the idea is maybe that methionine residue was in the structure. So that experiment allowed us to generate a footprint of the structured region of the TDP43 low complexity domain. And so what I'm showing you here is kind of histograms that correspond to individual methionine residues across the entire LC domain, low complexity domain. So residues 307 and 311 didn't have much O18, but you got to 322 and the ratio of O18 to O16 was quite high. The next residue right next to it, 323, also it was quite high. So the idea was those residues were protected from the initial oxidation with O16 H2O2. And so we did that in long polymers from hydrogels. We also did it in liquid-like droplets. And we also did it in living cells. And we saw the same footprint in all three places. And so we have a strong sense that the structure that we're studying in hydrogels and droplets actually is biologically meaningful because we see it in living cells. So the question is, where is that footprint? Well, I show below on the sequences of a number of these proteins. The region shown in yellow is the footprinted region, and it's the region that's absolutely most conserved from fish to humans, 400 million years of evolution. There's not a single change. It's the most evolutionarily conserved portion of the entire TDP43 protein. That's the region that has the molecular footprint. So it gets even better, and it gets better because of work from David Eisenberg. So David has solved the cryo-EM structure of this exact same region of the TDP43 low complexity domain. So he had three different structures that all shared a similar morphology right in this region shown in red. So from tryptophan 334 through proline 320, the structures were superimposable. Well, what was gorgeous to us when we saw David's paper is that dagger-like structure of around 15 amino acids, shown in red here, it corresponds precisely with the region that was footprinted, and likewise, of course, corresponds to the evolutionary conserved region. So here, I'm just kind of going straight from our footprinting data. Let's look at methionine residue 322, the most protected region of the footprint. That's this residue right here that's definitely within the Eisenberg structure. The next residue, methionine 223, that's the second most protected in the footprinting assay. That's also in the structure. And then these three methionines, 336, 337, and 339, that are slightly protected as a function of structure, they're right on the edge of the Eisenberg dagger structure. So we were elated. We were elated to see David's work, and we think we're slowly climbing up the mountain with this, and we're not alone. We now have other people, terrific scientists, who've kind of joined the fray. So how is this structure held together? Well, it's held together again. It's cross beta. It's held together by Pauling hydrogen bonds. So let's look at, at this kind of a schematic diagram of Pauling hydrogen bonds across a beta pleated sheet held together by hydrogen bonds. And so this goes back to 1952. Linus Pauling in his PNAS paper taught us about that, and he was exactly right. And so what we'd love to be able to do is to mutagenize, not to change our groups. That's what we've done for the last 30 years. Molecular biologists can mutagenize till the cows come home. You can change any residue you want to anything you want to. So if I wanted to change any of these R groups in the protein, that's a piece of cake. That's not what we want to do. We want to change the chemical nature of this hydrogen bond that goes between this peptide nitrogen and the carbonyl oxygen. So we don't want to change the R group. We want to change the ability of each residue to participate in this hydrogen bonding interaction. So you can't just mutagenize the main chain of a protein. You can't program E. coli to make you a protein that's missing that hydrogen bond. But fortuitously, we recruited Glenn Lissag, and right next door to me, I have a terrific peptide chemist trained in Tom Muir's lab at Princeton. So he also knows enzyme chemistry. And so together with Glenn, we've been able to attack this in a different way. So what we've been able to do is methylate peptide nitrogen. So in order for this hydrogen bond to form, it has to be an NH group. If we methylate that nitrogen, we've capped it. It can't do the hydrogen bonding. So what we've done is used peptide chemistry and enzyme ligation to walk our way along and systematically remove peptide nitrogens, or systematically cap peptide nitrogens, so we could ask the question, are single hydrogen bonds important for the formation of this structure? So let's go to the next slide that shows kind of the genius of Glenn and Xiaoming and Lily, the three people who have done these experiments over the last 2 years. So here's the TDP43 low complexity domain going from residue 262 to the C terminus of the protein. We want to study that, and we want to eliminate one hydrogen bond at a time across this region that we think is important for forming the structure. So how do we do it? So we make one protein in E. coli, that's fragment A, and by enzyme chemistry, we put a thioester at the end. The second part of the protein we make synthetically. So we can make a synthetic peptide, and we can make the synthetic peptide so that a particular residue bears a cap on the peptide nitrogen. So we can put that peptide nitrogen cap wherever we want to by making synthetic peptides. So that's the second part. The third part of the enzyme ligation is also made in E. coli. So we have three parts. We take this E. coli part, infuse it with a synthetic peptide by native chemical ligation. Once that's made, it bears the capped nitrogen residue, and now we can link that to the last fragment, the C-terminal fragment, again by native chemical ligation, and now we have our protein, but it bears two scars. It bears a scar here and a scar here that represent cysteine residues, but they plan things out beautifully, because what you can do is desulfurize these cysteines, and that turns them into alanines. And the way they planned the experiments, we already knew we had alanines at those two positions. So after curing the scars, we have a protein that's absolutely perfect, except for one residue, one nitrogen residue has been methylated. So this was a big risk for Glenn and Xiaoming and Lily, because who knew whether single hydrogen bonds would be important for the formation of these structures. So just 3 weeks ago or 2 weeks ago, we opened the envelope, and here's the result. So here are 24 proteins that are identical. Every protein is identical except for bearing a single nitrogen atom capped with methyl. So it goes from residue 316 to 339. And the question is, would any of these hydrogen bonds hurt? And so we performed our assays of phase separation, and sure enough, 8 of them hurt, but 16 of them didn't hurt. So it's not that all of them hurt, but about a third of them really impeded phase separation. And so the question is, where do those map? Well, those 8 variants where a single methyl cap on a peptide nitrogen hurt go from 321 to 329. And that's precisely the location of the dagger structure from David Eisenberg. It's precisely the location of the footprint. So we're really confident that we're on the right track, that sure enough, cross beta formation of this or a highly related variant of this structure is the basis of how TDP-43 protein, the TDP-43 protein is able to phase separate. Now, now we get to ask a really cool question. And the question is, by studying these things in such detail, so reductionist, paying so much attention to phase separation and biochemistry and peptides, is it leading anywhere? And I'm very hopeful that it is. And so we get back to this question of, when the RNA granule is going up and down the appendage of a neuron, how does it know to be translated? How does that granule unfurl at the site of an active synapse? And we have now an idea. And the idea is very simplistic. We know that active synapses are chock full of mitochondria. That's a definition of an active synapse. They're chock full of mitochondria. So our idea is really simple. And that is, in that environment, there's oxidation potential. Reactive oxygen species are made by mitochondria. If those reactive oxygen species can oxidize the TDP-43 LC domain in exactly this region and disassemble the interactions that I've shown you in test tubes, maybe that's the basis of how an RNA granule is unfurled at the proper place to facilitate localized synaptic translation. Now that's speculation. We don't know that, but by studying the chemistry of how these things work and go into the detail, we can at least envision a possible a hypothetical answer to this sort of question, which is at the heart of neurobiology, which to me is extremely exciting. All right, now I'm going to end with the last part of this talk that addresses human diseases caused by the mutation engendered addition of a single hydrogen bond. And I think this is crazy cool. I've never presented it before. This is the first time, so I'm real excited. So where did this come from? So I showed you how we introduced specific methyl caps to individually impede hydrogen bonding across this structure, but we also did a glycine scanning mutagenesis. And so here is, again, the TDP-43 LC domain. If we scan glycine residues across this whole region, the same region hurts. I mean, so that when you change the side chains, the R groups, that matters too. So don't think that it's only the hydrogen bonding that's holding things together. The side chains are also important. I don't want to go into that, but what I want to say is the absolute worst mutation, the one that hurt the worst, is proline residue 320. When we mutated that to glycine, it was an unmitigated disaster. The aggregation was incredible. So compared with all the other mutants, that was by far the worst. And in fact, if we change it to any residue, we change it to serine, we change it to leucine, any change of that proline was brutal. And so here's the liquid like droplets for the wild type protein. Here's the proline to glycine at residue 320. If we put those into cells, the wild type protein goes right to the nucleus, it behaves perfectly. This proline to glycine mutation, if we put that in cells, Xiaomin is found that it's exclusively in the cytoplasm. It can't go back and forth, it can't shuttle. So now let me go to the next slide and and look in David Eisenberg's structure and say, where is that proline? So this is a proline right here that I'm pointing to right there. And so why is that so important? Why, when that's changed to any other residue, why is it such a disaster? And let's go back to the chemistry of our amino acid side chains and realize kind of why proline is so unique. Well, proline is the only amino acid that can't do hydrogen bonding because the peptide nitrogen used by everyone else to do Pauline hydrogen bonding is occupied in this ring. And so there's no hydrogen atom. So there can't be any hydrogen bond at prolines. So they've long been known to block alpha helices, prolines block helices, and they block beta sheets. And the reason that they're incompatible with alpha helix and beta sheet is they can't do hydrogen bonding. And of course, hydrogen bonding is at the heart of both formation of helices and cross beta structures. So why is it that when we change that proline residue to anything, things get so bad with TDP-43? So in essence, we've changed it to glycine, and let's go back and forth. Here's proline, here's glycine, here's proline, here's glycine. So we've just changed that R group right there to glycine. And in the process of making that change, we've regenerated a hydrogen bond. All right, the idea is that that hydrogen bond, the absence of that hydrogen bond was important for insulation, that it wanted to stop this cross beta, this hydrogen bonding network, and the proline is stopping it. Yet if we change it to any other amino acid, we let that Pauline, the network of Pauline hydrogen bonds go too long. So, then we ask the question, okay, if we take the proline to glycine mutant, and now we cap the nitrogen atom, does it correct everything? And beautifully it does. So that terrible mutation is caused because it's taking a glycine residue that insulates and stops the extent of beta and insulates it to prevent it from extending. And if changed to any other amino acid, that hydrogen bonding network proceeds way too far and creates an aberrantly stable structure. So that caused us to think, well, maybe this is happening in other proteins. And so we started looking at the human genetics of neurodegenerative disease and other forms of neurological disease for mutations in proline residues. And so here's two papers that deal with proline 301 to leucine and frontotemporal dementia. Well, this is in the tau protein. Well, here's another another paper on tau that mapped a mutation to the exact same proline 301, but this time it's threonine. So you can mutate it to leucine, you can mutate it to threonine, and then it's terrible disease. So we go over and we look and found the same thing in H&RMP. So H&RMP has a low complexity domain. It has a proline mutation, and multiple patients have had the same proline mutation. Also in a neurofilament protein called NFL, there are proline mutations at two locations over and over and over. So there are a number of diseases where the disease-causing mutation is in proline residues. So now we can ask the question, can we fix those mutations the same way that we were able to fix the proline mutation, the proline to glycine mutation in TDP-43? So now let's look at this. We'll start with this neurofilament protein, which has proline mutations in its head domain. So the head domain is of low sequence complexity. The tail domain is of low sequence complexity. The middle of the neurofilament protein, as with all other intermediate filaments, is a coiled coil. So these mutations are not in the coiled coil, they're not in the tail, they're in the head domain, and over and over and over, they happen to either proline residue 8 or proline residue 22. The mutations in one family can be leucine, another can be glutamine, they can be anything. if you change that proline to any other amino acid, that's disease causing. So beautiful human genetic studies have showed that these foul up the intermediate filament. So the neurofilament can't form. So here's the normal neurofilament. If it bears this, proline 8 to glutamine, it makes a mess. It can't make good intermediate filaments. If it changes it to, if proline residue 22 is mutated, same thing, a disaster. So we wondered, what's the basis of these proline mutations? Let me just briefly tell you that the way you assemble intermediate filament proteins is kind of stepwise. You start by taking two coiled regions to make a dimer. That works perfectly well with a coiled region. Two dimers can get together and make a tetramer, and that works perfectly well with just the raw domain. But to go from tetramer to mature intermediate filament, you have to have the head domain. The head domain is not crucial for dimerization or tetramers, but to go from these tetramers to intermediate filaments, you have to have the head domain. And we and others have shown that, and it's because the head domains can form labile cross beta interactions. So we know that and we published that. But now let's look at these mutations in the head domain of the neurofilament protein. So they're in either proline 8 or proline 22. And so if we just study the head domain by itself, the wild type head domain behaves nicely. But as soon as you put in this proline 8 to glutamine residue, it's a disaster. It causes the head domain to interact way too tightly with itself. So if we look in the electron microscope, everything's clear here, but with that mutant, the head domain is a terrible aggregate. Well, if we methylate the peptide nitrogen associated with the glutamine, so we retain the exact side chain, but all we've done is blocked hydrogen bonding. Now we correct the mutation, no more mess. Same thing with the proline 22 serine mutation. It's a terrible aggregate if the head domain alone, but if we cap that peptide nitrogen, we cure the mutation. So it's not just in head domain interactions. We've done this with the intact intermediate filament protein. So again, with Glenn Lissak's help, we've used entein chemistry to take a peptide so that we can make synthetic peptides where we've methylated the nitrogen at the appropriate proline 8 or proline 22 residues. Then we can use entein chemistry, make an intact intermediate filament protein where we've done the same thing that we did with TDP-43, selectively methyl cap an individual amino acid. And when we do this, we look at intermediate filament formation with this simple mutation, P8Q, the intermediate filaments are terrible. But with that one methyl cap, we completely cure and make gorgeous intermediate filaments. So again, and the same thing happens with the P22 mutation when changed to serine. If we methylate that residue and prevent hydrogen bonding, we cure the ill effects of the mutation. So I think we've come to understand what's going on here, that these proteins need weak cross-beta interactions to perform their function. But if that interaction becomes too strong because of mutational effects as shown here, then biology does not work properly. So we've done the same thing with the HNRMP-A2 protein. It has a low complexity domain. This is David Eisenberg's structure, so it makes a cross-beta structure. It has a proline residue. If that proline residue is 298 is changed to leucine, you get bad droplets. It stabilizes this structure inappropriately. But if we go in and use entein chemistry and peptides that cap the peptide nitrogen associated with that leucine, it beautifully cures the phenotype. So I think we've traced it down in this case. And finally, we've done the same thing with the tau protein. So over and over and over, proline 301 in the tau protein gets changed. And it can be changed to threonine, serine, or any one of a number of amino acids. And if we make that peptide and study its aggregation, it's a disaster. That's a terrible mutation. And if we methylate the peptide nitrogen associated with that threonine residue, we completely cure the effect, not only in test tubes, but in living cells. So what I'm going to do now is wrap up and offer my thoughts about what I've been saying over the last hour. First of all, eukaryotic cells utilize thousands of different low complexity domains. They're all over the place. They're on all aspects of biology. I want to contend that certain low complexity sequences interconvert dynamically between states of labile, cross-beta structure, and random coil. And these allow for the formation of dynamic semiamorphous structures, including intermediate filaments. They're using this same chemistry. The structural state of low complexity domains can be regulated by post-translational modification, including phosphorylation. I have not shown you that today, but we've published on it, and I'm confident that we're on the right track there. But as of what I've shown you today, methionine oxidation and the control of the TDP43 LC domain self-association. And finally, missense mutations specifying neurodegenerative disease can change the balance of structural order to disorder in low complexity domains via mutation engendered addition of single hydrogen bonds. And to me, this is telling me that this 10-year undertaking that we're still on the right track. We're trudging away, but I think we're going in the right direction. So there's still unanswered questions. I want to kind of end with these unanswered questions. So why is it that the low complexity domains are between 2 and 4 times the size of their cross beta cores? So as I started with fuse, it makes a cross beta core, but 70 or 80% of the low complexity domain is not folded in that core. So why is it that it only uses a small proportion? And what is the use of the unstructured sequences that are flanking these cores? Do they have any purpose? So we don't know. How are the cross beta cores chosen from these low complexity sequences, from the quasi-repetitive LC domains? In other words, how do they know to follow the Amundsen rules? How do they know to form the appropriate exact structure, the biologically appropriate structure? We don't know. The sequences are gibberish looking. It's hard for us to see why they would choose one region versus another to form their core structure. The third question is, does oxidation-mediated reversal of the TDP43 LC domain itself association, does that facilitate synapse localized translation? That's an idea that I've thrown out. We don't know that. And yet if it were true, it would really have us focus on a question of fundamental biological significance. And I hope we're on the right track. I hope that might be true, but we don't know that to be true. That's just an idea. And finally, might an understanding of the science someday lead to informed avenues for therapeutic intervention? By understanding what's going wrong, or will it lead to some way of thinking about how we could help to attack these devastating neurological diseases? So with that, let me end with acknowledgements. And so a number of people have worked with me here at UT Southwestern over the last decade on this science, and they're listed here. We've had a lot of wonderful collaborators, but I want to highlight today Masato, Xiaoming, Lillian, Ben, Tu, and Glenn. These are the people who really helped us push the new science that I showed you in the second half of my seminar today. So the last thing I'll do is tell you that these two men are the people who taught me how to do science, Don Brown and Joe Gall. And my time at the Carnegie, together with Don and Joe, taught me how to do science. Don taught me how to not go horizontal. When I'd made a discovery, keep going vertical, have courage, move forward. Don't just milk a project, but leave it for others and go on to the next thing and try and climb the ladder, try and go vertically. Joe taught me rigor and scholarship, not to be narrow, but to think more broadly and to know more than just the focused details of molecular biology, to think more broadly as a biologist. So these two mentors. I am incredibly dedicated to these two men. And with that, I'll stop and be happy to try and answer any questions. Thank you for your attention.
Thank you so much. Thanks, Dr. McKnight. That was great. It was such a pleasure having you. Yeah, we have a few questions here in the Q&A. I will read them out loud. First question is for Homa Lin. High temperatures might cause protein folding. Kids who are exposed to high temperatures for a period of time, it might impact their IQ. Is there any finding which part of the brain or brain cell might play a role in this?
You know, I just don't know enough science or medicine to answer the question. It's a good question, but I can't answer it. I'm sorry.
All right, and we have a question from William Hale. Is it easy to see, or it is easy to see how the cross beta architecture you showed for, is it FUS? I'm sorry, I'm not familiar.
Yeah.
Could yield the unbranched filament like you saw in the hydrogel EM. Is this different in the droplets?
That's a good question. So what's the difference in the protein in the hydrogel versus the droplet? Well, unquestionably, the hydrogel, the cross beta interactions create long polymers composed of thousands of subunits. And in the droplet, instead of having thousands of subunits, the interactions form maybe only 2 or 3 or 4 subunits. So we've observed the same structure in the droplets, but the interactions that form the droplets, instead of being thousands of units in the subunits in the polymer, they're only a handful. So the polymers are way, way, way shorter. That's what we believe to be the difference between the two.
All right, another question from William. Why does the low complexity of the peptide matter for forming the cross beta structures? Is low complexity somehow intrinsically predictive of phase separation?
No, so the question is, as I understand it, is of what value is the cross beta interaction from these low complexity sequences? What's the use of that? And our interpretation is that structural interaction, transient as labeled as it might be, is the basis of specificity, such that a low complexity domain knows how to interact with itself and not just mistakenly interact with any other low complexity sequence. So the cross beta forming region is a bona fide molecular structure that has specificity built into the structure. And we think that's the utility of the structure, is to create interaction specificity so that a granule or an intermediate filament can properly form by use of these unusual low complexity domain sequences.
And I'm going to read this question from Corey Handwerger. What about the interaction between LC domains and different proteins within the same biological structure?
Yeah, I mean, let's consider an RNA granule. An RNA granule, it contains messenger RNAs. It contains, probably multiple different RNA binding proteins. And so if we're thinking about the low complexity domain of TDP43, we argue that it knows how to see itself, but we don't have any understanding how the low complexity domain of TDP43 might interact with other components of the granule. Does it touch other low complexity domains coming from other RNA binding proteins? We don't know. So we're at a very early stage. We kind of have a sense of how things work, but we're at the earliest stages. So I just can't answer the question.
And we have a question here from Craig Holbrook. How will these understandings end up being translated to medical invention therapies?
Well, if I knew, if I had a good sense of that, I would have already formed a biotech company to make use of that information. So I don't myself see at this point how this understanding is going to better enable us to tackle these diseases. But I will say that many people in the field, this phase separation field is become very, very popular. And there are lots and lots of very good high profile scientists in it. And 2 or 3 biotechnology companies have already been founded over the last 2 or 3 years that have raised upwards of hundreds of millions of dollars. And so the money's flowing. I don't see how our knowledge at this point is going to lead to therapeutic intervention. But the good news is money's going into it, and a lot of smart people are in the field now. So I hope and trust they have better ideas than I do.
Catherine Thompson says, very exciting work. She asks, can you speculate on how this could be used to develop therapies for, I guess that's the same question, for neurodegenerative diseases? We can move on, unless you have something to say again there.
If it's a Catherine Thompson who was a postdoc with me at Carnegie, I want to say hi to her. Hi, Catherine.
Dan Wasserberg asks, are the cross beta cores hydrophobic? Sometimes, always?
Yeah, that's a good question. So the low complexity domain, the cross beta core, the fuse low complexity domain is exceedingly hydrophilic. As I showed, it only has one hydrophobic residue within the entire interface, which is a proline residue. By contrast, the short dagger fragment, whose structure has been resolved by David Eisenberg, it does have hydrophobic amino acids. So methionine is hydrophobic. So I think it will vary from example to example. And so I don't think there's any standard answer to that question. So it's too early. We only have a few examples.
And we have a few more questions here from Stephen DeLuca. The proline mutations seem like they would be dominant. Have you tested in vitro or in vivo how much mutant protein it would take to disrupt normal function of wild type protein?
Okay, so the geneticists indeed know that these are autosomal dominant mutations. So that's true. And the question is, have we done experiments that would be compatible with that genetic dominance? And yeah, I think everything that we've seen so far would be compatible, but we have not specifically attempted to address that point.
And we have a question from Diane Shakes. When a proline is mutated and you get a bad droplet, what's going on in those abnormally shaped droplets? Is it becoming a solid?
Yeah, that's a very good question. So when you mutate proline to any other residue, as I've said, that regenerates the capacity for Pauling hydrogen bonding. And so our assumption is that you've allowed for cross beta interactions, Pauling hydrogen bond interactions, to be extended further from what their normal restrictions are. And yet we don't have any structures of mutant polymers formed from proline mutants. And so that's our belief, is that hydrogen bonding is extending beyond where it normally should, and that might lead to more stable structures, but we have not nailed that. All we know is we see, very rampant aggregation of the proteins. It could be tau, it could be TDP43, any of them. When they have that proline mutation, we see very profound aggregation. What that aggregation is at the molecular level, we don't yet know.
Charles Hunter asks, are there low complexity domains in prokaryotes? What functions might they perform?
Yeah, in fact, there are way fewer. That's a very good question. And so if one does, appropriately computer searches in prokaryotes for low complexity domains, there are way, 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 way fewer. So there's, stripped down versions, their proteome is stripped down and largely devoid of this kind of gibberish stuff that is, that was the topic of today's seminar. But they're not completely gone. So Lucy Shapiro had a very nice paper on a low complexity domain in Caulobacter. A young scientist at ETH in Zurich, Maria Hondel, has studied an RNA binding protein in E. coli that has a low complexity domain that behaves in this way. And so there are a few of them in E. coli. And for the ones that have been studied to date, they seem to be responsible for similar sorts of interactions, probably to localize proteins in appropriate puncta subregions of the bacterial cytoplasm.
And I'll ask 3 more questions from the group here. And apologies to anyone who didn't get their question answered, or if you asked a question after you're getting your first question answered, I will download the transcript and email them to Dr. McKnight, and we can get the answers to you separately. So Barbara Graves says, great science. Can you move the Dagger domain to other proteins to drive novel behavior?
Again, Barbara, hello. She was a postdoc at Carnegie also, if it's the same Barbara. And I've talked with Ben too about, trying to move the TDP43 LC domain into yeast to replace a methionine-rich LC domain in yeast that he studies. My gut feel is that's not going to work because there's a lot more sophistication of interactions, but that's a good question. And so where might you want, an oxidation sensitive intermolecular interaction, and maybe you could design this to be transported to another protein to endow it with oxygen sensitivity.
All right, 2 more questions. Kariyash Karamian asks, could you localize a protein to the mitochondria to see if it can get preferentially oxidized, result in aggregation?
Oh, that's a very good question, because our idea is that you get in proximity of mitochondria, and that's where you're going to be susceptible to oxidation because the reactive oxygen species being made by mitochondria. So that's all speculative. That our idea as to how it works. And so she's asking exactly the right question. Can we do experiments that show that proximity to mitochondria enhances, susceptibility? And I think the answer is probably yes. You put some sort of mitochondrial targeting signal on your protein, you get it in the vicinity and ask the question, is it more susceptible to oxidation in that environment? We haven't done that, but I could imagine thinking of ways to get at that. So very, very good question.
And last question, and I think this is great to end on, and I apologize if I get your name wrong, but Hongru Liu asks, could you elaborate on going vertically instead of horizontally in science, as Don told you?
Oh, okay, that's a good question. So in essence, you work real hard on something, and let's say, what I did as a postdoctoral fellow, what was called linker scanning mutagenesis and of the thymidine kinase promoter. And I worked really hard with Bob Kingsbury, and it took 2 years to scan these mutations all across the promoter of the thymidine kinase gene. And luckily, it really worked out well. We developed a map of the promoter that was one of the first maps to tell us about how a eukaryotic transcriptional regulatory domain would work. And having kind of worked that out, I thought, well, I know how to do this now. And since I'm real good at this, and so it's a hot area, I just got a paper in Science. I'm going to, do it on this next promoter. So, and Don would say, that's horizontal. You've already done that. Don't go leftwards and horizontally to another promoter. You already know how to map it. Go vertically and go see if you can purify the transcription factors that make your gene work. Well, vertical is hard. I mean, I'd never done biochemistry. I'd never purified a transcription factor. And so Don said, don't go horizontal to things that you already know how to do. Go vertical and challenge yourself to do something you've never done before. That's what I mean by vertical versus horizontal.